Hello, everybody. This is Ask a Washington Beekeeper. I'm Margot Buckles. I'm a volunteer with the Washington State Beekeepers Association. We started these Ask a Washington Beekeeper videos so that we could provide information and education to people who don't have access to a club or other forms of education. I'd like to introduce our speaker tonight. His name is Kevin Oldenburg. He has a PhD in biochemistry. He is the former president of the Washington State Beekeepers Association. And this evening, he's going to be talking about how the media gets it wrong when it comes to science, especially as it relates to beekeeping. So, Kevin, go ahead and take it away.
Well, thank you, Margot, for the introduction. And let me start sharing my screen and we can jump into the presentation. So here we go. Do you see this screen?
Yeah, just give me just one moment. Yes.
Excellent.
And
Okay,
if you want to, yeah, okay, you're good to go.
great. Thank you, Margot. So as Margot said, I have a PhD in biochemistry. I actually got my degree back in 1990, so it's been quite a while. I've done significant work in biochemical research, and I was also the head of drug discovery at DuPont Pharmaceuticals. And a number of years ago, I became the president of the Washington State Beekeepers Association, and I was the president for, I think, 3 or 4 years, something like that. Um, and actually, this talk is one I gave back in February of 2018 at our local association at the West Plains Beekeepers Association. And the reason I gave this talk at the time was because there was a lot of media attention in terms of colony collapse disorder and neonics. And as a scientist, I was very interested in what was the actual science behind the link between neonics and colony collapse disorder. So I started doing some research into that correlation or causation, and actually looked at the scientific papers that made that initial assessment. And so what I want to do tonight is give you an idea of where that science came from, how the media jumped on it, and how they actually ended up getting it quite wrong. Now, if I can get to the next slide, why is it not letting me move? There we go. So I want to start out by basically with a simple sentence. Science is not a set of results, okay? Science is really a method. And whenever you're looking at science, there's good science and there's bad science and there's mediocre science. And even sometimes very good science, you don't realize when you devise your method that certain aspects of that method aren't quite correct. Okay. And so, for example, scientists many times will present a result and then they'll publish that result or they will present the result at a conference. Other scientists will look at, basically the result itself, the method they got to that result, and an argument ensues. Almost invariably, an argument ensues. Scientists love to argue with each other. And in fact, you can argue very vehemently with another scientist and 10 minutes later end up having beer with them as one of your best friends. It's not unheard of for scientists to simply be at a conference and be screaming at each other because they both believe their results, or they believe the method was wrong, or they're just like to argue in general. So we, as a group of professionals, we simply like to be very critical of other people's work, and hopefully we use the same criticality in our own research. So what I'd like to do is start by basically defining what the difference is between a law, a theory, and a hypothesis. Because you see this happen a lot, that people, they'll say, oh, that's just a theory, that doesn't mean anything. Well, let's start with a law. A law basically describes a natural phenomenon based on repeated observations. For example, you can have the law of gravity, or the laws of thermodynamics, or the law of motion, which basically says that force equals mass times acceleration. Those are laws. They're simply things that repeated observations show us to be true. And there's really no way that you can use those laws to predict anything else. A theory, on the other hand, explains not only the facts as we know them, but also allows predictions of what should be observed if the theory is true. Examples of that are evolution, general relativity, and the Big Bang. I mean, I don't think you'll be able to find a scientist, at least a reputable scientist, that would ever argue that evolution is not true because it's been shown in the lab. Anybody who's worked with any type of bacteria knows that you can basically evolve bacteria very quickly in the lab. And the nice thing about the theory of evolution is that it allows you to predict what may happen in the future based on a set of criterion or a set of circumstances in the natural world. Now, on the other hand, there's another thing we call a hypothesis. A hypothesis is simply an idea that proposes a tentative explanation about a phenomenon or a narrow set of phenomena observed in the natural world. An example of this in current work would be something like varroa mites transmit viruses that are detrimental to bee health. Okay, that's a hypothesis. We think that might be true. And what a scientist will do then is we will design a set of experiments, basically a method of how we're going to test that hypothesis. And upon testing that hypothesis, we can then determine whether or not it's a valid hypothesis or not. If it's not, then you'll usually come up with another idea and begin testing that. If it's true, then what you'll try to do is recruit fellow scientists to reproduce that work that you've done and expand upon it. So let's take an example of basically back in the early 2000s, I think it was, we had an issue with colony collapse disorder. And from colony collapse disorder, what we got basically a survey that was conducted by the Bee Informed group from basically 2006 through 2016. And what they did is they went out and they asked a group of beekeepers, basically managed hives. They were really asking, unfortunately, my screen, I can't see this very well. I wonder if I can move this. Yes, I can move it. Okay, good. They asked, for example, what is an acceptable winter loss? What's the total winter loss you've seen? And then what's the total annual loss? And basically what they were looking at here, as you can see in 2007, for example, 2006, 2007, they said the acceptable loss was about 15%, but their total winter loss was in the low 30% range. And you can see in 2015, 2016, that the acceptable loss was up a little bit, maybe a couple percentage points. But the total winter loss was down a little bit, but the total yearly loss was up. So go to the next slide. You have to remember now that science is not a set of results, it's a method. If the method's wrong, the results are useless. So this is a classic example of how a group like this can really screw things up, especially when you report it to the layperson. So this survey was sent out to 5,756 beekeepers in the United States, and they present that in the data. So they say they sent it out to basically 6,000 beekeepers. So as a layperson, you would think, wow, that's great. They sent it out to 6,000 beekeepers, right? Well, there's two main problems with this on the face of it. The first is that there's about 2,000 commercial beekeepers in the US and 210,000 hobbyist beekeepers. Now, the problem they have is that they didn't report on the number of returns that they got. They said they sent it out to almost 6,000 beekeepers, but they didn't say how many beekeepers returned that survey, or were those surveys returned by commercial keepers or by hobbyist keepers, okay? So collectively, these beekeepers managed about 400,000 colonies in October of 2015, which represented about 15% of the US's total bee colony population, which on the surface of it sounds pretty good. 15% coverage is pretty good. An estimated 28% of the colonies managed in the US were lost in the 2015-2016 winter, which represented an increase in losses of about 6 percentage points compared to the previous year. But it was close to the 10-year average. So right there, that was an interesting piece of data from this survey, because there was really no report of a major colony collapse problem. That is, we had about the same number of colonies before and after the initial reports of colony collapse problems. Just over half of the survey respondents, about 60%, experienced winter colony loss rates greater than the average self-reported acceptable winter mortality rate of 16, 17%. Now, the number of colony losses to me has always seemed a little strange because if you think of the colony as an organism, individual bees are not the organism. The colony is the organism. And as we know from as beekeepers, any colony that overwinters is very likely to swarm. So if you even take a reasonable number of 80, 90% of the colonies that overwinter that swarm, if we didn't have comparable losses in colonies, we would be up to our eyeballs in bees in a matter of a few years. So that's why it doesn't surprise me at all that we have fairly high losses of colonies year after year. Now, interestingly, I was told by some beekeepers who have been doing it for many, many decades that before Varroa and some other issues we've had, that colony or beehives tended not to swarm quite as often. So that leads me to believe that potentially because of stressors like Varroa, that the Varroa pressure may be causing colonies to swarm more often. That's a hypothesis, and that's something that would have to be tested. Anyway, so one other piece of interesting information is that about 1% of all beekeepers, which are commercial keepers, manage about 86% of all the hives. 99% of beekeepers are hobbyists, and we only manage about 14% of our hives. So a report like this that you saw in the Bee Informed Network is really fairly meaningless unless they give you the background information to know who returned the forms and what type of beekeeper were they. So, for example, I would expect commercial guys to have much lower losses, or winter losses, than hobbyist keepers, simply because that's their profession. They're going to be much, much better at it than we are. So back in about, let's say started in about the mid 2000, around 2010, thereabouts, a little after. There was a huge neonic debate in the US. And in fact, we were seeing things where uh, the government was talking about banning all neonics. And neonics basically they're a nicotine-based insecticide, which is an interesting class of insecticides, by the way, because the tobacco plant basically produces nicotine for the simple reason that it prevents insects from eating the plant. All plants are trying to do this, but the only difference is that nicotine happens to be, give people a high, and people get addicted to taking nicotine. But anyway, so scientists found that, well, if this is a class of chemicals that can basically prevent insects from eating a plant, and actually the neonics themselves are good insecticides, maybe they'd be a good compound to use in agriculture, because it would prevent insect predation on plants. So a whole category of those insecticides have been developed by various agricultural companies and screened very intensively prior to them hitting the market, and they've been on the market for a number of years. But then in the mid 2000s, like around 2010, I think 2009, we started to see colony collapse disorder. And no one really understood what was causing this colony collapse disorder. It was a term that was coined, and there was never any really good data to show that there ever really was a true colony collapse problem. There were a few instances of colonies simply disappearing out of a hive, and no one knew where the bees went, and all the bees would be gone. But there was never an association. First of all, it wasn't very widespread. It wasn't like, 5% or even several percent of the managed honeybee colonies ever exhibited colony collapse disorder. It was very, very limited. Yet it got really an inordinate amount of media attention. And so some groups started to do research to try and figure out why or what was causing this perceived problem. And one of those groups was basically out of the Harvard School of Public Health. And they presented the first publication on the effects of neonic insecticides and colony collapse, that is, the destruction of all the bees in a colony. And it was done by Cheng-Shin Liu and others. And it was published in the Bulletin of Insectology. So, how does a scientist evaluate a research paper? Well, when we see a paper like this, there are a number of things, usually about 5 things that any scientist is going to look at. The first is, where was the data published? What journal was it published in? And there are multiple tiers of scientific journals. For example, at the topmost tier are journals like Science and Nature. If a publication gets into Science and Nature, it basically makes any scientist's career, because very few scientists ever get published in those two journals. The scrutiny on the research that's published in there is intense. It has to be groundbreaking research, and it has to be of the very highest quality. You're talking probably the top 0.1% of all science ever gets published in those two journals. Below that are journals like I have like Cell, and several others that are, there's probably a handful of 8 or 9 different journals. PNAS, which stands for the Proceedings of the National Academy of Science. And there's about maybe 8 or 10 journals that are tier 2 journals. And those journals are almost as good as the top tier journal, but they don't have quite the same prestige. But you can pretty much count on anything that's published in those journals as having been very highly peer reviewed in a very critical manner. So things that are in those journals are usually very, very accurate. Not that things aren't wrong there or get changed, because that's just part of science, but they're usually very, very well done. And then you have things like 3rd and 4th tier journals, which are, I've published in them as well. It's basically, you want to be able to get your research out there. You think it's good research, but it may not be of widespread interest to, the general scientific community. The review process is a lot shorter and it's not quite as intense. Then the thing you look at is this the first publication in a new area, or is it a verification publication? So usually what happens is, if you publish something that's of interest to a number of scientists, and you're the first publication, other scientists are going to go out there and try and either prove or disprove what you claim in your publication. And so when you look at it, is this the first publication? If it is, you'll take it with a little bit bigger grain of salt. Then the 4th thing you're going to look at is, were the methods sound? That is, did they use good science? Was it, were all the things that they did following the scientific method? And can you find any fault with what they did? And finally, then, is their data logical and internally consistent? Do they draw conclusions based on their data? And are those conclusions accurate based on their data? So are they not drawing inferences for things that they don't have data for? So we go back and look at this publication, it violates many, many of these rules. So first of all, if you think about it, where was the data published? Well, it's published in the Bulletin of Insectology, probably, I would say at best, a 4th tier journal. So that's one where you don't get a lot of intense review. And you have to take everything in that with a very big grain of salt. Then what are the credentials of the authors? The first thing you notice here is that, so Liu is in the Department of Environmental Health at the Harvard School of Public Health, Landmark Center West, Boston, okay? The next guy, Kenneth Warchol, is the Worcester County Beekeepers Association. And Richard Callahan, they don't even list his affiliation. So right there, for me, would be a very big grain of salt, because not a person on here is an entomologist. None of these people really have the credentials for doing bee research per se. You have a beekeeper, but that doesn't mean they're a trained researcher. Then if you look at, is this the first publication? Yes, this is definitely the first publication. It's not a verification, so we don't know if the results can be replicated or not. And then, were the methods sound? And we'll get into that in a little bit. But there's one interesting thing these people did in this article, in this journal. And what they did was they looked at the environmental concentration of neonics in plants that are usually being protected by those insecticides. And what they did for the first 5 weeks of their study was they fed their bees that concentration of neonic insecticide in sugar water, basically inside their hive. And so the bees would be eating this fairly low level of neonic only as they needed it out of their sugar water. And then after 5 weeks, they increased the concentration of the insecticide about 40-fold. So at that point, it was way beyond the LD50 of that compound. Now, LD50 stands for lethal dose 50, and that's the dosage at which about 50% of the organism will die if it consumes that compound. So they were about, I believe it was 40 times over the LD50. And then, oops, I hit the wrong button. And then, is the data logical and internally consistent? Well, based on what they did in their experiments, they could not draw the conclusion that neonics were causing colony collapse disorder. And we'll get into that in a minute. I'll show you why. So this single publication caused an entire flurry of basically other publications and comments in layperson magazines and journals. So, for example, from the July issue of Better Planet, they call it a bee apocalypse. Can we save honeybees from colony collapse disorder? From Slate, honeybees at a crossroad, drought, pesticides, and disease. It's not a good time to be a bee. And then from the Daily Telegraph in the UK, keepers warn of Bee-mageddon after France authorizes controversial insecticide. And then another one, Bee Apocalypse Now. Scientists discover what's killing the bees, and it's worse than you thought. Bee-mageddon threatens U.S. with food disaster. Bee-mageddon, pesticides kill over 10 million pollinators in Brazil. I thought that one was kind of cute because, as you know, there's 60 to 90,000 bees in a colony. So if you killed 10,000 pollinators in Brazil, that's a whopping 125 to 150 hives. And probably that many are killed every year in traffic accidents. So let's really look at the data and see what's happening to see, first of all, was there ever an issue with varroa mites or neonics? I mean, we know there's a problem with varroa mites for a number of reasons, but so let's look. So in 1987, varroa mites first entered the United States. That's the first documented case of a mite in the US. And on this graph, what we're showing is the total number of bee colonies in the US in thousands. So in 1987, there were about, what's that, 3,250,000 hives. And then B shows the year that colony collapse disorder started. As you can see, there was, in the first few years after Varroa, the number of managed colonies in the US dropped. But upon the use of basically the implementation of using neonics in the environment, the number of bee colonies remained relatively stable for the next 10 years. If you look at the number of, and this is from the USDA statistics, and I'm sorry, the USDA and the statistics from Canada. If you look at the number of managed beehives in the US, when colony collapse disorder was identified in 2006, you can see after that in both the US and Canada, the number of managed beehives remained relatively stable. And same is true of in the European Union. What you're looking at here is neonics were widely used in Europe. There were 4 different versions of neonics, 4 different insecticides in the market at that point. And they were all widely used starting in about 1998. And you can actually see in Europe, after they started being used, the number of managed beehives actually increased over that time. So you could say, okay, so what's going on? Well, if you look at the number of colonies, the change in the number of colonies over time from the late 80s through the early, through 2010, you can see that year on year, there are times when the number of colonies increases and times when the number of colonies decrease. And the increase in colonies and the decrease in colonies have no correlation whatsoever with the introduction of varroamite or the introduction of neonics. Really, all it is the law of supply and demand. If the growers need more colonies, they will order more colonies for pollination services with their commercial keepers. The commercial beekeepers know how many colonies they're going to need, starting with the almonds down in California and then throughout the US. And what they will do is they will basically split their hives in order early in the season in order to optimize their profits. So they will increase the number of hives that they're going to have if they have need of the services. If they don't need the beekeeping services, they will get rid of the hives because they don't want to maintain them because that's an additional cost. So there's no correlation whatsoever between the number of hives, managed colonies in the US, and colony collapse disorder or the introduction of neonics. So that publication I told you about is really interesting. So what happens is when a scientist makes a publication like that or submits a publication, other scientists will read it. And what they will do is they will then try and determine whether or not that data is any good. And so what they will do is they will either do their own publications or they will do their own research, and they will also a lot of times speak publicly on that, usually at a conference of other scientists. So here are a couple examples. So in the State of the Universe, do neonics hurt bees? Researchers and the media say yes, the data do not. Basically, a new landmark study provides plenty of useful information if only we could interpret it accurately. So there was interesting information in their first publication, the Lou publication. And then from the University of Illinois, entomologist Mae Berenbaum, who chaired the National Academy of Sciences in 2007 National Research Council study on the status of pollinators in North America, called it effectively worthless to serious researchers. The experimental design and statistical analysis are just not reliable. And finally, this is from, I forget this last guy. It said it's surprising that these colonies lasted so long, given the stratospheric quantities of insecticide Lou pumped into them for 13 weeks. Oh, that was Jonathan Getty. And basically what he is saying is that, like I mentioned earlier, that for the first 5 weeks, they used a concentration of neonics that would be seen in the environment, that a bee might be exposed to when they're out pollinating flowers. And then for the next 13 weeks, they increased the concentration by some 40-fold, I believe, which was so high that it's basically, there is no way any colony could ever survive that. It's just incredible that the colonies even lasted as long as they did when they were pumped full of such a high level insecticide. So that's why this publication is really a junk science, because this is a classic example of a scientist changing their method because for the first 5 weeks, they didn't see any change in the colonies. So these colonies were being fed an environmental concentration of neonics that they would have seen in the environment, and there was no discernible change or effect on the colony. Well, they didn't want to see that. They wanted to see an effect, so they increased the concentration of the insecticide to such a point that they would see the effect. Let's see, why is this not changing? There we go. So Richard Culls, who's a prominent entomologist in the state of Connecticut, said, when used properly, there is intriguing evidence that neonics may actually improve bee health in some circumstances. Hints can be found, ironically, in Lou's own data. Basically, his paper raises red flags because he used 2 separate dosing regimens as the experiment progressed. During the first 4 weeks of his study, the bees were fed concentrations of imidacloprid that, as it turns out, were in fact field realistic. At 3 weeks into testing using these concentrations, the health of the bee colonies was positively correlated with exposure to the imidacloprid as measured by the number of capped brood cells. In other words, the bees appeared healthier. So in the first 3 to 4 weeks of the experiment, the bees were actually healthier by one measure of their experiment. Rather than continue the experiment with these concentrations, Dr. Liu inexplicably increased the dosages for the last 9 weeks of feeding by 40 times. Okay. So at that point, he's basically increasing the concentration to such a level that all of the bees are going to die. And so what was his conclusion? That basically giving the bees basically the neonic caused the colony to die. Therefore, colony collapse disorder is caused by neonics. Okay. That's like saying, you put arsenic in their water, and amazingly enough, they died. Therefore, arsenic is causing colony collapse disorder. Okay. The problem is they're poisons. Any poison is going to kill those bees, and it's a function of concentration. It's the same. The same is absolutely true of any drug that, as a human, we take, which is why pharmaceutical companies, like the ones I worked at, spend so much time working on dosing regimens. Because if you get the dosage correct, it's actually therapeutic. And if you get a dosage wrong and it's too high, it can actually become toxic. So Liu's work is clearly biased and sensational, said Richard Culls. It is horrendously incompetent. This is just hogwash. We'll all pay the price for bad research, which we did because his research was bad. It got picked up by the media. The media splashed it all over because it sounded sensational. It hurt the agricultural companies that had developed these neonicotinoid insecticides, and it also hurt the farmers because they were, these really good, relatively safe insecticides were, for a time, actually pulled off the market. So in a 2013 PLOS One study, a team led by Van Engelsdorp and Jeffrey Pettis studied the real-world impact of 35 pesticides, including three neonics, by examining hives from 7 major crops. Intriguingly, bee health improved, although the results would need to be confirmed with follow-up research. So there were at least 2 studies, the initial study and then the study done by these guys, that showed that low levels of neonics actually tended to improve the health of honeybee colonies. So in this study at the time, so this is 5 years ago, and I haven't looked at it since, remained at that time the only lab research to date that evaluated how real-world pollen pesticides blends affected honeybee health, which would be really interesting to do. So they found nosema. Another thing that was interesting was they found that nosema infections were greatly reduced in hives treated with field level concentrations of neonics. They don't know if that's a cause and effect or if it's just an abnormal, an example of a statistical outlier. Which also reminds me to tell everyone that one of the big issues I have with bee research is that the number of experimental subjects that are used is much, much too small. So having spent a lot of my career doing drug research for humans, drug research typically works like this. Scientists like myself, biochemists primarily, will look at a chemical pathway in an organism like a human and try and figure out what disease does that, or what effect does that pathway have on a specific disease state in a human. Once we know that, then what we'll try to do is find compounds that will either enhance or inhibit that particular pathway. Once we find those drugs, and they go through extensive animal studies to show, at least in animals, that they do what we think, that they're safe, they don't cause other side effects as much as we can, we then go into a human trial. The very first human trials are usually done on upwards of 500 people who are healthy adults. They get the drug at the concentration that the scientists and researchers think will be effective for the disease they're trying to treat. If the company sees and the research shows that there's no detrimental effects on healthy humans, they will then go into a limited clinical trial on upwards of about 5,000 individuals that have the disease. Of those people, they will get the drug at various concentrations, and they will see how they tolerate the drug and whether or not it actually affects the disease. If it works on those 5,000 people, they'll go to an expanded clinical trial, which could be in upwards of 20 to 50,000 people that they'll get enroll into a trial, and basically do the studies on them to see if there is a problem at that stage. Now, there's an example of a drug that we worked on. It was a great drug. Um, the problem was when we went to a much, much bigger basically study of several thousand people, we found that about 1 in 1,500 people, it caused a very serious side effect. Basically, it caused them to rupture a high proportion of their red blood cells, and it would kill them, which is why we do extended studies. The problem with bee research in general is that you have to remember that the colony is the organism, not the individual bee. And so, like in a drug study in humans, when we study a drug, we have to do thousands of individuals because each individual is very different from the other individuals. And we need to find if there's any, is it general, does the drug work for basically everyone in that population? Or for some reason, do you have a subset of people that it works very well in? So, for example, Pacific Islanders, it may work great in because their genetics are somewhat different than Europeans. And so that's my issue with bee research is most of the time, the scientists simply don't have the resources to use the number of colonies that they would really need to get a statistically significant output or statistically significant data from their research. So they're drawing conclusions on data that many times they don't have enough data points to really draw that conclusion from. I will skip that slide. So in conclusion, I want you to remember that science is a method. If the method is wrong, the data is junk. And as a scientist, we really don't believe anything until it's been replicated many times. If we see a publication come out once, it might pique our interest if we're in that area, and we will typically try to repeat that experiment, or we will try to basically extend the research that was done there and see if we can expand upon it. If we can't, then we will more than likely conclude that the original data may not have been very good. But again, if it works, then we can expand upon it. And it's not unusual for having two scientists, present two different hypotheses that are very, very different, argue vehemently against each other's research, and argue for years on end about each other's work. And as you see that what happens is, they start way apart. And as time goes on and they do more and more experiments, and other people may join into the experiment, doing experiments as well. When they were, miles apart, in years, what happens is their research tends to merge and come together, and they tend to draw the same conclusions eventually. The other thing I want you to remember is that media loves sensational stories. So if you hear it in the media, the only reason it's out there is because it's sensational. They know it's going to get reads, it's going to get talked about, it's going to sell papers, it's going to sell newspapers, it's going to sell time, on the internet, or it's going to sell time on TV. And so they love sensational stories, but a lot of times the data behind a sensational story isn't all that accurate. And then finally, lay people like simple explanations to complex problems. Most of the things that scientists are working on are extremely complex and very difficult many times to explain in ways that are very simple and quick and easy for people to get the gist of. Scientists need to definitely get better at that. So anyway, that's the end of the talk, and I'll turn it back over to Margot for anything that she has to add.
Well, I don't have any, there aren't any questions here on my end. Ellen, do you have anybody on Zoom?
I do not see any here either.
Okay, well, I think then that we're done for this evening. Thank you very much, Kevin, for the very interesting talk about how science works and how the media gets it wrong.
Thank you, Margot.
And we'll see everyone next month for Ask a Washington Beekeeper. Thank you so much.